This is a project conducted by Marine Science Protector students Sebastian Peralta and Ala Lucier under the vision of our instructor Kendall Romani. The area that the project aims to take place is at the rock pool at airport. The place where we conducted the research is a man-made pool. It was dynamited so it could create a pool for the community to have a place to swim. Greetings everyone! This short documentary is about the biodiversity of microalgae within the upper fringe of the rock pool adjacent to the Amana Kabua Airport. The form of methodology that we used: first we started out with the measuring tape to measure current water level. Keep the tape about 2 meters above the water level and measure to point. It must start from the upper fringe of the rock pool till it reaches 2 meters. This will be the area of study in which everything will take place. To measure the rock pool full width, measure to the full end, the starting point, of the rock pool. Secondly, to get the percentage of the microalgae, the quadratic chart will be used. The quadratic chart must be placed at 2 and count the number of algae within each 25 square boxes in the quadratic chart. Remember, each box in the quadratic chart must be filled half or all in order for each one to be counted. Lastly, the circumference of the rock pool is 166.42 feet. This was taken after you took the width of the rock pool, which is 53 feet, to determine circumference or perimeter of the circle, multiply 3.14x radius x2. The research questions: what are the different types of microalgae assemblages and the physical habitat structure of the upper fringe section of the rock pool ecosystem at the airport? The aim: determine the biodiversity of microalgae and the geological composition of the upper inner fringe section of the rock pool ecosystem. A lot of factors were seen during the observation, as the place is very unique in its own way. The survey was about the biodiversity of the microalgae present in that location. The area around the rock pool, which is the upper fringe, 3 species of microalgae were present in the surrounding upper fringe: sea lettuce, Phaeophyceae, and Chlorophyta sensu. There are 6 points section in which we identify the percentage and the types of microalgae within the upper fringe of the rock pool. The depth of study was 2 meters, as it was the appropriate amount for us students to conduct regarding health and safety. There were some limitations that we encountered throughout the project. For example, we were limited on the proper equipment that would have made the whole data collection process easier. No scuba diving suits and tanks to stay longer underwater. We were not able to go deeper without the suits and tanks. Also, the reason we chose to work within 2 meters is because we weren't fully equipped with the proper tools to collect the samples. 2 meters was the amount we chose as it was easier and safer for both of us. Furthermore, because we were working on a short time frame, it was required that we do what was more convenient and effective. Also, the rock pool ecosystem as a whole was full of different types of species. For example, we were able to identify the different microalgae living around the fringe of the reef at the airport rock pool. Overall, we were able to find the percentage of the 3 species that we found at the rock pool: sea lettuce, deathman finger, Padina pavonica algae, and gutweed Ulva intestinalis. There are other species that we're seeing within the upper fringes. While there are still large gaps in our knowledge of these curious sea cucumbers, these observations challenge the traditionally held notion that they are solitary, non-social animals, only using their body patterns to avoid or startle predators. In addition to the diverse array of marine life rock pool support, they are of particular interest from an ecological research and education perspective as well. They act as mini dioramas of larger ecosystems, and due to their accessibility and small size, provide a natural laboratory where an entire ecosystem can be studied with ease. Because they come in many different shapes and sizes, they allow researchers to understand how different habitats shape marine communities. To conclude, the project was a success. This was one of the hardest wrecking projects that we ever did. We learned a great amount from this project. The rock pool itself was really unique, as it is an ecosystem in itself that does not get a lot of outside connection into it. For example, the rock pool and all the creatures within it are what is circulating inside of it. No other creatures or outside beings are able to enter it. With that said, it's a whole different ecosystem that is cut off from the rest of the other ecosystem. Thanks for watching our short documentary. Big thanks to Mr. Kendall Ramini for giving a truly inspiring vision in creating this video. Once again, thank you. From the true protectors of marine science.